Yeah, thanks, Tatiana, for giving the overview on the activities of the BioFoundation. Now it's my pleasure to kick off the second day of our academic symposium on AI in life sciences. So I'm happy to introduce Katarzyna Bozek, who's currently a professor at the medical and computer science faculties of the University of Cologne in Germany. And her research is focused on applying deep learning methods to biological imaging data, all of that with an interest toward detection of tissue pathologies. Before her current position, her career took her to 5 different locations in 3 countries, so working in about as many fields and spanning both human health-related projects and also evolutionary and environmental topics. So just like Bayer, Dr. Bozek has been active in health and crop science, if you wish, or at least adjacent to crop sciences. So after her undergrad studies of computer science, she did her PhD thesis at the Max Planck Institute of Computer Sciences in Saarbrücken, working on evolution of host virus infections, particularly the HIV virus. After that, she did a joint postdoctoral fellowship with the Max Planck Society and the Chinese Academy of Sciences in Shanghai. And during that period, she studied the evolution of the human brain. Results of her work were not just published in the academic literature, but also covered in popular media such as New York Times or National Geographic. And after that postdoc, she obtained her first group leader position, this time in Japan, the Okinawa Institute of Science and Technology. And here she first started working on applying modern deep learning methods to biological imaging data. And in this time, in particularly trying to track members of honeybee colonies from a simple video of the colony. And now in her current position, Dr. Bozek is focusing her research again on topics relating to medicine and human health. And today she will talk to us about a current project regarding prediction of skin cancer recurrence. So please join me in welcoming Dr. Bozek.
Yeah, hello everyone. Thank you for the invitation, Florian. It was we're quite interested to be here. Actually, this living in this academic bubble that we do, seeing the startup scene here at Bayer is really interesting for us, and I wish for more of such interfaces between actually what we do and the industry. Right, I guess I have to swap slides. Right. So like Florian said, I'm at this cross-section between computer science and the medical faculty in Cologne. And my group is working sort of mostly on image data. So image data is our main focus. And we develop, apply, sort of fine-tune deep learning models to address a multitude of medical problems, sort of across branches of medicine. So we work on oncology and pathology data, obviously, but also in nephrology, eye medicine, neurology, so you name it. And we're really driven by this sort of the developments that happen in deep learning, and we're curious to now apply those developments also in the field of biomedical research and medicine itself. And today I'm going to talk to you about a project that I particularly like. It's this very nice combination of a real world medical question and a problem, but also that we managed to develop some really cool computational solutions that sort of the nerd people like we really are interested in. So like my group members particularly. So the story is about skin cancer, one particular type of skin cancer. So there's those different cells in skin, and what we're focused on here are the squamous cells, and that's where the tumor actually emerges. So it's a type of non-melanoma skin cancer. It's actually quite common. Sort of many people will develop it, especially at advanced age, but most often that tumor can just basically be removed by a surgery. Except a significant number of patients that will develop metastasis, right? So for those people, they will recur in some years, and actually then their prognosis is very bad. So that recurrence is a very sort of negative event. So as you can imagine, it's really important to identify those patients, yeah, and to offer them treatments ahead of time before it's too late. So how the skin is diagnosed, where here are such histopathology slides, right? So the skin surface is on the top of those slides, right? And somewhere in there is the tumor. And that's what the pathologist or dermatologist will look at in the diagnosis. And right, so one of them will belong to someone who will recur, the other one to someone who will not. And so the question is, right, how do we distinguish those two types of patients? Yeah, and this slide, just to give you a zoom in, is composed of millions of cells. It's not billions, right? So there's a lot of information in there. And what I need to mention is that this particular staining is some kind of chemical staining typically used in histopathology, which makes visible key morphological elements of that piece of tissue, right? So we will see cell nuclei, we will see their shapes, we will see fibers, and some major types of cells can be recognized just based on this simple staining. Yeah, so exactly. So that's exactly the question we want to address here. Right, so we worked with other clinical centers to collect as many of this type of images as we can, and importantly also where we have that recurrence information. So that is not so obvious in a country like Germany, where sort of those medical systems are not centralized and we might just lose track of patients. But eventually, we have 3 cohorts from Bonn, Cologne, and Munich, which amount to roughly 500 patients. And I will describe you briefly the model that we developed to address this question, as this is actually what we like to talk about the most. So those slides that we're looking at here are really big, right? So there can be 100,000 of pixels in one dimension. So we cannot just train one simple network to classify, right? Those will be too big for a GPU memory. And also, right, we need them at the right resolution, so we cannot really shrink them, crop them, right? We need the full thing. So the typical approach to those digital pathology problems is this multi-instance learning, where this image is cut into little tiles, right? This kind of little images, which we pass through some sort of linear transformation or little network here to represent them then with numbers. Yeah, so this way we already crunch the volume of information here. Instead of pixels, we represent every little patch with numbers. And then we have some sort of model here that aggregates that information. So we might have, let's say, 100,000 little patches here. And in one of those patches, it might be just in one of the patches that there is the key information, right? There is tumor here, and that's the type of tumor. And so this aggregation element here needs to be sort of sensitive for that, right? So we need to pick up that information in that large bag of little images. And then comes the classification. And that's the general sort of family of such methods, yeah? And now what we did, so there's a pool of them, you can just take them from the shelf from a publication. Now what we did, which we were quite proud of, was to do exactly this. So we start with cropping the image. We have the list of patches. We represent them with our vectors or embeddings, we call them. This, so I marked this with colors here. This pink or violet, depending on the screen square here, is some sort of pre-trained neural network. Yeah, and then we introduced that sequence shortening mechanism here. So that's multi-head attention for those who are experts here, which basically allows us to go from this list of 100,000s or sometimes 1,000, it can be variable length of little embeddings, to crunch it into a fixed and short length sequence. And so why do we need that? Well, the general idea here is that our predictive model, what we use here is a pre-trained language model. Yeah. So what the language models really require, such that you can use them, is that your list of tokens, your list of words that you input is of some fixed length, yeah, fixed dimensionality, which was not possible for those big slides, right, in pathology, because, right, first of all, they are huge, and second of all, they might have varying length depending on the size of that piece of tissue. And here we introduced that and this white square here is a trainable element of our model, right? So this we learn. So we have this learned mechanism to shorten that sequence. You can think of it as a compression mechanism. And we have a language model that we don't train, which is why it's in pink. And then we have just some classification component in the end. So all in all, we take a pre-trained language model, which we know that is a powerful thing, right? That sort of when we published, there was like a maybe a year and a half ago that we actually published this method. And we didn't really use those large language models here. We wanted something that can fit on a GPU. So so the really surprising thing here was that we could just take something that has never seen images. I know that now it's maybe more obvious to everyone that vision and language models can intersect. Back then it was just a language model that has never seen images. So that which we took here, and without training that model, we actually reached a good accuracy, which I show in this little table. Like the bold numbers here are our results, which we show that beat other models in some benchmark comparisons. But in addition, what we argue here for is that because we don't train our language model, we don't train our embedding element, just this compression part, we have a very lightweight model, and it takes very little to train it, and it's actually quite performant. Anyway, so that's our computational work, and now we applied exactly this to predict recurrence and non-recurrence in our skin cell problem. Yeah. So what we did first was we thought, okay, let's train our model on the colon cohort, and then we will see sort of how far we can go on the other cohorts. And as you can see here, that's worked with quite a variable accuracy. So it seems like we do quite well on the colon cohort, but for the other cohorts, it's really varied and sometimes random, sometimes good. We actually don't know why, which is quite a common problem in this type of models. So then what we do next, we did this federated scenario, right? Where we don't share the data between the centers. We just sort of update our model weights separately on each cohort, and we just aggregate them centrally, but sort of we don't mix our data sets necessarily in one place for training the model. We just do it in this kind of federated and sort of secure manner. And that actually is what solved the problem. So we have like a really performant model. I mean, to give you, I don't have that curve here, but I mean, there's some clinical markers that people know matter for this disease progression. But with those markers, we can only go up to 0.65 accuracy, let's say. And here we're up to 0.80 something for most of our cohorts. So that's great. So we have a really performant model, and that's not the end of the story. So those So we also wanted to gain some insights, sort of what is that predictive element of those slides that allow us to know what will happen to the patient. And there's different ways to introduce this interpretability to deep learning models. And we have tried several of them. In the end, we end up using this one, which is sort of a gradient-based method where we compare the image with, let's say, a blank image. And we see sort of which pixels, which regions of this image pull the prediction in which way based on the gradients, roughly speaking. Yeah. So what this method promises, right, that it will highlight elements that are that the model uses to make a particular prediction. So if it's a cat, right, it's going to highlight the cat in this image. A dog will highlight a dog, right? So that's what we do. We calculate those cumulative gradients between our target image and just the plain image. Then the question is like, yeah, what next? And I just wanted to sort of squeeze it in here on this slide that we also have segmentation models for those particular, not tumor, but the tissue type, right? So there's many different segmentation models out there. What we did ourselves was to fine-tune a model that would particularly work on cells that are present in skin tissues, right? So this is our the types of cells we can identify on an example piece of slide, and sort of we can now segment those entire slides and know for those millions of cells what each of them is. Right, and this is what we call this attribution score, which is basically this gradient-based thing. So if this slide belongs to someone who has recurred, a progressor, then the red parts here will mean that this patches of the image pull the prediction in the direction of progressor, right? So this is how we interpret this. The red parts here is where the information is that the model uses for the given prediction. Yeah, and we can do this for progressors and non-progressors. And how we do it then is that we invert the score for the non-progressors, so to basically have sort of the same score for all the slides. Yeah, so that would mean that positive means it pulls prediction towards progressor, negative for the non-progressor. So we have that score for all the patches for all the images. And then we basically look what is in those sort of high attribution score patches on both ends of that distribution, right? So both in the direction of non-progressor. So what makes you sure that like actually you're safe and you will not progress, versus what are the regions that are responsible for progression. Right, and then again, how can we do this on a large scale, right? Because we have those hundreds of images, we have those billions of cells and millions of patches, most probably. We can segment them. We have the attribution score. So we needed some smart way of aggregating all of those elements and doing statistics. Yeah, so given the segmentation information we have, we can calculate all of those things, right? We can calculate how many cells we have in the high attribution score patches, what is their morphology, we can use that spatial information actually that we have, which is also very important for tumor microenvironment, right? So what are the distances between different cells? And one thing that we did was also this joint count analysis, and I'm going to just explain briefly what that is. So imagine we have this hypothetical country that has departments, and this country will be divided in some way, right? There could be some language differences, or maybe they vote differently, right? So this country could be divided this way, it could be divided this way, right? So here, the gray and the white parts of the country have actually. So the gray parts of the country don't share borders, yeah? So they're absolutely isolated. So there's more black-white borders in this distribution of that voting preferences. It can be also like this, which we would say is roughly random, right? There is some clusters and there's some dispersed labels here as well. So this is what we call joint count analysis, right? We basically very simply count the borders between black and white, the joints between black and white parts of this map. And in a clustered scenario, this number will be lower. In the dispersed scenario, this will be higher. And that's actually a very useful measure that we can very easily, in a sense, quantify, and that can tell us a lot about those spatial interactions in that piece of tissue. Yeah. Right. So what we do, I'll maybe skip those technical parts, but yeah, we binarize that segmentation map. We do a Voronoi we split those regions with a Voronoi algorithm, and we count the joints. Okay, so what characterizes high attribution score patches? Where we did all of those statistics, right? We looked at all those features that we quantified, and we compared them in the sort of two ends of the attribution score distribution, yeah? And so what you have on the right side of this plot is the distribution of tumor cell count in the patches that are predictive of progression, and the green parts that are predictive actually of non-progression, yeah? And we found actually several features that are actually very, very significantly differ between those two groups, yeah? So one of them is tumor cell count. What it means is that the information, whether a patient will progress, especially if the patient will progress, that information is not in the tumor. That information is actually somewhere out there. And if the information whether a patient will not progress lies within the tumor. This is what this sort of two distributions tell us. Then sort of the joins ratio, right? So there's, since sort of the predictive information about progression is not within the tumor itself, there will also obviously be more of those joins between tumor cells and the rest in those patches. Yeah, so those interactions might be predictive, might be important piece of information. Then we looked as well at things like distance of lymphocyte cells to other cells, and this distance seems to be lower for the progressors, surprisingly. And it's things about sort of aspects of morphology of cells where we also found differences. So in particular, the predictive patches for progression seem to have larger and very sort of irregular shaped cells. Right, so here is to give you, so we spent last several weeks sort of looking at those distributions and histograms. I'm just going to flash a few results here in front of your face. It's not easy to sort of gather this into a coherent story, but I think we're about to really have it. So this is an example slide of someone who has progressed, right? The tumor is somewhere here. It's this sort of darker part of the slide. And here's how this features distribute in that tumor area, right? So the like I said, the yellow part here is where we have a lot of tumor cells, right? So that's the tumor region. Then just outside of it, right, the middle histogram here is where the stromal cells lie, right? And they interact, right, with other cell types. And very importantly, the attribution score histogram shows us that the predictive patches are really on that border, on the outside of the tumor. One more example of someone who is a non-progressor. So the tumor is on the left side of the, depends how you look at it, yeah, on the left side of this slide. And here is where like the tumor cells interact with other cells. And just wanted to point out here that, again, this attribution score really is not on the borders of the tumor. It's really also inside of the tumor. Yeah. And so the story that we are sort of now coming up with is that the pattern that is really more typical for the non-recurrent patients is more like on the left here, right? So that's where we have really that split, sort of that the tumor is more of a cluster and there is less of this interface between the healthy tissue, whereas recurrence is more like the one on the right. And that's where this information, the predictive information lies, where we need to look for it. And similarly, right, the type of cells, the morphologies that we will see in those regions that are predictive or recurrence will look more like the ones on the right, right? So these morphologies are more irregular, they're elongated, they are sometimes not really convex, versus compared to those regular and round-shaped cells you see on the left. Right, so that's the story that I wanted to show you. Basically, it's really cool that it seems like we can now use machine learning AI to really go beyond a simple diagnosis and based on histopathological slides. So it's not just about detection and classification, but it seems like we can also predict into future, and that's really cool. Right, we have good models to do that. Yeah, and that information is not necessarily within the tumor. So we need to look at the tumor microenvironment, which seems to be a sort of a common knowledge now. And in particular, in this type of tumor, those cell-cell interactions and on the outside of the tumor and cell morphology is what we find to be meaningful for those patient outcomes. I just wanted to acknowledge the people who have done actual work. So Juan is the one who's the brain behind the predictive model and who did the whole analysis. Lucas did the segmentation model. And our medical partners is Doris and Johannes from dermatology. And these are our collaboration partners from other clinical centers. Yeah, so thank you.